I now call to order the Society's 2,426th meeting in the 149th year since its founding on March 13th, 1871. Good evening, everyone, and welcome to another installment of the Fall Lecture Series produced by PSW Science. These are unusual times, and because of COVID-19, the Society is bringing these meetings to you via Zoom from locations all around the world, rather than our usual home in the John Wesley Powell Auditorium of the Cosmos Club in Washington, D.C. Tonight, we will hear about recent work on aging and the possibilities for extending healthy life and for extending the lifespan. Our speaker, Eric Verdin, has spent much of his career working to understand the processes of senescence and how to slow them down. I'm Larry Milstein, President and Program Director of PSW Science, one of the oldest scientific societies of Washington, D.C. PSW Science is a 501c3 nonprofit education and science organization whose mission is to provide a forum to exchange scientific ideas, further scientific understanding, and encourage scientific inquiry. This lecture is being recorded and will be posted to the PSW Science YouTube channel, where it will join over 140 other recordings of PSW Science lectures. We invite everyone to take a look and explore these presentations. And if you like what the Society is doing, we invite you to become a member and make a contribution through the PSW Science website, www.pswscience.org. The Society is grateful for the sponsorship of the 2020-2021 lecture series by the Policy Studies Organization in cooperation with the American Public University, and a generous donor who wishes to remain anonymous. We're also grateful to the sponsor of today's lecture, PSW member Tim Thomas. Thank you, Tim. Before we turn to the lecture, it is the tradition of the Society to welcome new members and read minutes of the previous meeting, summarizing the previous lecture. I am pleased to announce the following new members. Amar Chalan, a student studying computer science and engineering, interested in organic chemistry, physics, and astronomy, who learned of PSW by searching the internet. R. Scott Kemp, a professor at MIT, interested in nuclear measurement, energy resources, biosecurity, and public policy, who learned of PSW from PSW member Richard Garwood. And tonight's speaker, Eric Verdin. We welcome them to membership. Recording Secretary James Heelan will now read the minutes of the 2,425th meeting and the lecture by Davey Smith. His lecture, COVID-19 Treatments and Trials, is available on the PSW Science YouTube channel. James, the screen is yours.
Thank you, Larry. Good evening, everyone. On September 25th, 2020, by Zoom video conference broadcast on the PSW Science YouTube channel, President Larry Milstein called the 2,425th meeting of the Society to order at 8:02 p.m. Eastern Daylight Time. He announced the order of business. He did welcome new members. The recording secretary then read the minutes of the previous meeting. President Milstein then introduced the speaker for the evening, David Smith, professor of medicine at the University of California at San Diego. His lecture was titled, COVID-19 Treatments and Trials, Treatments to End a Pandemic. As of Smith's lecture, there were approximately 32 million confirmed global SARS-CoV-2 COVID-19 cases and almost 1 million global deaths. The objective of Smith's lecture was to answer the question, can we treat our way out of a pandemic? The reproductive rate of an infectious disease is calculated by multiplying the number of contacts an infectious person makes by the probability of transmission per contact and the duration the infected person is infectious to others. For SARS-CoV-2, the reproductive rate is between 3 and 5 secondary infections in a completely susceptible population. Current efforts to reduce the number of new infections to 0 are aimed at all 3 elements of the reproductive rate equation. Quarantines, physical distancing, and social bubbles are intended to reduce the number of person-to-person contacts. Mask wearing reduces the probability of transmission. Antiviral treatments and vaccines are being developed to decrease both the probability of transmission and the duration of infectiousness. Smith then specifically focused his lecture on the development of antiviral treatments. Antiviral treatments will be most effective against SARS-CoV-2 in the early stages of infection, when viral is highest, while infected persons are showing mild to no symptoms. Initial treatment efforts began by researching whether existing drugs could be repurposed to treat SARS-CoV-2. Examples include ACE inhibitors, camostat, hydroxychloroquine, and remdesivir. In early 2020, data from cell cultures showed hydroxychloroquine to effectively inhibit viral growth. Early human tests produced similar results, and the FDA gave emergency authorization for hydroxychloroquine to be administered to infected persons showing symptoms. Before Smith and the National Institutes of Health could complete a trial on the drug, reports emerged that hydroxychloroquine was dangerous to patients, and the FDA revoked its emergency use authorization. In retrospect, Smith said, because antiviral treatment is most effective in early-stage SARS-CoV-2 infections, administering hydroxychloroquine in late-stage infections likely had minimal effect on the virus. A small sample size study from the Henry Ford Foundation showed a higher survivability rate among people who received hydroxychloroquine early compared to those who received a placebo. Remdesivir was developed to treat SARS-CoV-1, called Middle Eastern Respiratory Syndrome, or MERS, and has been repurposed to treat SARS-CoV-2. Gilead Pharmaceuticals owns the drug and conducted a trial that showed patients treated with remdesivir experienced a shorter recovery time than those given a placebo. Because remdesivir has a short half-life and must be administered intravenously, Smith said it has limited use outside hospital settings. Developed to treat Ebola, EIDD-2801 interferes and causes the virus to make fatal errors when attempting to duplicate its RNA. EIDD-2801 proved effective against MERS and is now in clinical trials for use against SARS-CoV-2. Scientists are also working on new drugs, including antibody therapies. Current efforts include collecting plasma from a recovered person and administering it to infected persons. Smith said that there was a terminated small sample trial which produced early data that convalescent plasma treatment could be effective in patients with severe but non-life-threatening cases of SARS-CoV-2. Smith and others are working on monoclonal antibodies. Some recovered people produce highly effective antibodies which can be identified, purified, and expanded, and used to treat other persons. This strategy has been effectively implemented to treat Ebola and HIV. Another current approach by SAB Pharmaceuticals, transplant a human immune system into cows, expose the cows to SARS-CoV-2 to prompt a polyclonal antibody response, which can then be used for human treatment. Smith said, severe inflammation in persons infected with SARS-CoV-2 is caused by immune system overreaction. Immunomodulators and anticoagulants are being explored to treat such cases. Smith is the chair of the US federal government's Operation Warp Speed. Active 2's objective is to rapidly and effectively and efficiently evaluate multiple potential therapeutics for COVID-19 in an outpatient setting to prevent them from requiring hospitalization. If the study determines an agent will prevent death or hospitalization through 28 days after study entry, the FDA will approve the drug. Smith is optimistic and believes that yes, we can treat our way out of the SARS-CoV-2 pandemic. The speaker then answered questions from the online viewing audience. One member asked about using colchicine to treat the virus. Smith said the drug will probably be used to treat late-stage infections, but is still under study. Another member asked about a small study on calcifediol in Spain. Smith said the Spanish study was excellent and that larger studies are needed. He said, but for the delay between the initial outbreak in China and the initiation of rigorous studies, many of the treatment questions would already be answered. The robust question and answer period then continued with detailed questions and answers regarding various clinical trials and details about the virus. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:28 p.m., President Milstein adjourned the meeting. The temperature in Washington, D.C., 18 degrees Celsius. Weather, showers. The number of concurrent viewers of the Zoom and YouTube live stream, 86. And views on the PSW Science YouTube and Vimeo channels, 359. Respectfully submitted, James Heelan, Recording Secretary.
Thank you, James. The minutes will be posted to the website in due course. Please send any corrections or comments on the minutes to Corresponding Secretary Robin Taylor at correspondingsec@psw.org. And we now turn to tonight's lecture, Coming Revolution in Aging. Will we live to be 150? And it's my pleasure to introduce tonight's speaker, Eric Verdin, who is speaking to us from across the country in Novato, California. Eric is president and CEO of the Buck Institute for Research on Aging and an adjunct professor at the University of California, San Francisco and University of Southern California. Previously, he was associate director of the Gladstone Institutes and held faculty positions at the University of Brussels, the NIH, and the Pickauer Institute. Eric's research focuses on the role of epigenetic regulators in the aging process. His laboratory was the first to clone a family of enzymes called HDACs, which regulate histone acetylation. Eric studies how metabolism, diet, and small molecules regulate the activities of HDACs and sirtuins, and how they affect the aging process and its associated diseases. He is an author of more than 210 scientific papers and an inventor in more than 15 patents. He is the recipient of the Glenn Award for Research in Biological Mechanisms of Aging, a AAAS Fellow, and has been elected to the American Society for Clinical Investigation, the Association of American Physicians, and Belgium's Royal Academy of Medicine. Eric earned his MD from the University of Liège. All questions will be fielded after the lecture during the question and answer period. Eric, the screen is yours.
Thank you, Larry. I'm delighted to be here. I want to thank you and your members for the opportunity to speak here today, and I look forward to this interaction and to a great discussion with all of you. So let me share my screen. So I chose a title which is a little bit provocative, and I hope that by the end of the talk I will be able to answer this question. So we are today at a historical inflection point, both from a societal and scientific point of view. And the presentation that will follow is going to be discussing both what is happening from a societal point of view in terms of aging, but also what has happened during the last 20 years in terms of our understanding of aging. And we are truly at an inflection point. It's been said that this decade will be looked back at a time when many of these key questions really came together. And I hope that you will leave this lecture with a sense of optimism in terms of what lies ahead for all of us. Why is there an inflection point? I think best way to illustrate what is happening in terms of our population, in terms of aging, is to really look at Japan from 1920 to 2010. The reason I chose Japan is because they are probably about 20 years ahead of where we are going. Germany is in the same situation. The US is a little behind. So what do I mean by where we are in terms of population? On this graph, I'm showing you the population in million of Japan over the last 90 years since 1920. And what is illustrated is the fraction of the population that is age 15 to 64 versus 65 or older versus 0 to 14. And what you can see has happened today in 2020 in comparison to 100 years ago is that the fraction of people who are 65 has been rapidly increasing. That is an important change in society, but it is nothing next to what is predicted to happen in the next 40 years in Japan. That is, the fraction of people age 65 is going to increase, to continue to increase, such that by 2060, it is estimated it will represent close to 40% of the population. Now, I just ask you to pause for 1 second and to envision what a population would look like when more than 40% of people will be above pension age. This has enormous implications in terms of retirement, in terms of our whole system of entitlements, the way we actually have them in the US and in Japan. And so this is what I think is, to be honest, really should be of concern to all of our governing bodies. It is obvious that the current system as it works today cannot work under such a fraction of population being older than 65. Another way to look at this is if we look at a percentage of the global population, this year for the first time, the number of people aged 65 or plus has become larger than the number of children who are less than 5. And a bit of a humoristic way to look at this is that for the first time this year, we are selling more diapers for adults than we are selling them for babies. So we have, across the world, a rapidly aging population. US is, because of immigration, is actually not in a worse situation, but the prediction is that we will eventually go to where Japan and Germany and other countries are today. Now, another way to look at this data is to actually compare the age at death for people at 2 different time points. One is 1900. You can see here at that time, there was a large infant mortality. A lot of children died in their first years of life. And the peak of mortality later was around 70 years old. Now, of course, in 2016, infant mortality has decreased very significantly, but we've also seen a dramatic shift at the age at which most people die. You can see the peak today in the US is around 88. In France, right now, it is 92. So really a dramatic change is at the age at which people die. What this graph also illustrates in orange is a phenomenon that actually no one expected, is that at that age, once you passed the age of 71, a number of chronic diseases appear that are really strictly linked to aging. We call these diseases the chronic disease of aging. And they're illustrated here. And most of you will be quite familiar with them, either because you are experiencing them personally or because a member of your family, especially parents, have experienced them. And so these diseases are atherosclerosis that manifests itself by either via heart attack or stroke, eye diseases such as glaucoma and macular degeneration, many cancers, obviously not all cancers. There are some cancers that are specific to childhood, but many cancers actually show a very large increase during aging. Osteoarthritis, osteoporosis, type 2 diabetes, Alzheimer's and Parkinson as 2 major neurodegenerative diseases. Sarcopenia, which means a loss of muscle mass, and finally hearing loss. Now, the relationship between aging and these diseases is best illustrated on my next slide, which shows the number of deaths per 100,000 individual in the population per year from each of these conditions that I just mentioned: heart disease, cancer, stroke. Actually, here we've also added emphysema, diabetes, kidney disease, and Alzheimer's. And you can see an incredible increase. I'll point to the fact that the scale on the left is a logarithmic scale, indicating that there is really a dramatic increase of these diseases as we age. What has not been clear is why, and how are we going to actually deal with this problem? Because what it means today is, as we live older, we also are living sicker. And when I've asked the question to people, would you want to live to 150? The response is universally no. And the reason is that we justifiably associate old age with disease and suffering. Some of the points that I want to make today is that the two are not necessarily associated. And there's light that from the science that we and others have conducted that suggests that we can uncouple aging with some of its diseases. Not forever, but at least for a while. So what is aging? I love to share with you this analogy, which is that of a 1940 pickup truck, which is shown here. And you do not need to be a mechanic to actually identify this truck as old. It is rusting, lost its wheels, it's been sitting unattended for a while. However, I'm going to show you on the right side a picture of a truck which is exactly the same age, a 1940 pickup truck. And this one is actually in the same condition as when it rolled off the assembly plan. So this actually throws a wrench into the idea that we really understand what your chronological age does not have much meaning. That is, both of these trucks have the same chronological age, but they are obviously aging very differently. And so it goes for humans. We all age at different rates, and this eventually manifests itself in the fact that many of us are dying at different ages. So I want to introduce here the concept of biological age. Think about biological age, the equivalent of what we are seeing on this picture. The truck on the right has a very low biological age, and the truck on the left is actually quite aged biologically. And we know the same thing happens in humans. So what is aging? It is, in fact, what is allowed the truck on the right to maintain in this state is it was in the hand of a collector, which constantly repaired it and fixed it so that every piece as it became older was restored to its original state. And so it goes for us. Aging is thought today to be the consequence of a loss of maintenance and repair and this slow accumulation of unrepaired damage. And in effect, what the science of aging is dealing with today is the realization that as we age, throughout our life, actually, we are constantly being repaired. We are subject to a number of assault that includes irradiation, from cosmic rays, that includes the weight of gravity, that includes DNA damage, that includes protein misfolding. All of these processes are accumulated with damage and are actually repaired quite efficiently by our organism. So one of the concepts that is emerging in the field of aging is this idea that by activating these maintenance and repair systems, we should be able to mitigate the aging process. And much of the data that has been generated in animal models in the laboratory actually support this model. So let me shift to actually some of this data and to show you what has been accomplished and what have we learned in the last 100 years about aging. Perhaps one of the biggest discoveries of the last 20 years, and one that actually has generated a lot of excitement, is the realization that genes control lifespan. Now, if we think back about what we knew before, it must have been obvious that genes control lifespan. We know different organisms which are defined by their genetic material live with very different lifespan. If you compare a mouse to a whale the mouse lives about 3 years, the whale lives about 200 years. Obviously, this is a process that is defined by the genes of the mouse and the genes of the whale. So, however, there was a discrepancy or a problem, is that no genes had been identified by mutation that could actually increase lifespan. And back in 1988, it started with the work of Tom Johnson, but was rapidly followed by the work of Cynthia Kenyon and the work of Lenny Garante at MIT and UCSF, respectively. Were able to identify mutations in a variety of small organisms that actually were not deleterious. This is the vision that we had of a mutation until these papers were published, but mutations that were actually associated with an increase in lifespan. And I want to share with you an experiment from Cynthia Kenyon here that follows lifespan of this little worm shown here on the right. It's called Cinerabidis elegans. It is about the size of a comma, and it lives normally 21 days. So this is the normal lifespan. So the way these experiments are shown, we start with 100 worms, we follow them over time, and as the day passes, the number of living worm decreases, so that by 23 to 24 days, most of the worms actually have passed. Out of this data, we can define a maximum lifespan, but we can also define an average lifespan. Now, what Cynthia Kenyon showed is that a single point mutation in a gene called DAF2 was associated with a dramatic increase, not only in maximum lifespan, but also average lifespan. The worm actually lived 3 to 4 times longer. What was even more remarkable is that combining this mutation with removal of the gonads, of the sexual organs of this worm, actually led to a 10-fold increase in lifespan. You know, this is the type of data that when reported generated an enormous amount of excitement, because it highlighted the fact that these worms had a lot of, I'll use a term which is perhaps not scientific, but life potential. So there was a lot of life in these worms that actually was not realized. What was even more remarkable is that these worms showed an extension of every single stage of their lifespan. So even though they lived close to 10 times what a normal worm would live, this life was actually had a longer growth period, a longer mature period, and a longer old age period. So this is the type of data that generated, again, excitement in the lay press, and led some people who I would say should have known better to claim that humans were going to live 1,000 years old. Now, I should say that while we also have many of the genes that have been identified in the worm that increase lifespan are conserved in humans, one of the things that we have observed as we move up the evolutionary chain is that the gains in lifespan become smaller and smaller. So at this point, there is nothing in the data that would suggest that we're going to be able to make humans live 10 times longer. And I frankly I'm not sure that this is actually a desirable outcome. So this is the effect of genes, which is quite remarkable. Today, if I go back to this slide, actually we have identified more than 500 genes in the worm, C. elegans, that actually control an increased lifespan. So this observation is not an exception. It is actually quite easy to increase their lifespan via genetic manipulation. Obviously, changing gene and introducing mutations is an important tool in biology. This is what allows us to move forward quite rapidly, but this is not something that is being considered for humans, at least not for the foreseeable future. So there's a lot of interest, therefore, in trying to understand what is the relative role of the environment, lifestyle, versus genes. And the next experiment that I'm going to show you suggests that a lot of the lifespan can also be determined by lifestyle environmental factors. And interestingly enough and remarkably enough, this experiment that I'm going to be discussing was conducted by Clive McKay back in the early 20th century. What he did is a lifespan experiment in mice. This is shown here. Mice, the normal mice here showed a lifespan of about 36 months. They live about 3 years under the laboratory conditions. And what he observed is by restricting the amount of food that these mice ate, he could, if he restricted their food by 25% in comparison to what these mice would eat, the mice live longer. And the more he restricted the food, actually the longer the mice lived. And you can see here again, a close to 50% increase in lifespan in these mice. Obviously, if food intake is further restricted, the mice end up suffering from malnutrition. And so based on this, one can derive actually a link between nutrition and lifespan, which is quite interesting, and it is shown here. And remarkably also is the fact that this curve is not linear. It's actually a bell curve. And I want to highlight the fact that many of the interventions that we look at in the aging field actually manifest themselves with this type of curve. So more is not always better than just enough. And I'll illustrate this in 1 second. So if you look at this slide here, looking at the food intake and the lifespan, obviously, if the animal is not eating, there's malnutrition, there's no growth, and there's no life. As the food intake progressively increases, you can see lifespan increasing. It turns out that most of us actually are eating a little bit too much for a maximum lifespan. And this is ad lib, meaning ad libitum. This is what most of us do. The calorie restriction, malnutrition is obviously here. Calorie restriction would be this peak of nutrition that leads to the longest life. I should say also that there is a cost to calorie restriction, which is that it is also associated with a decrease in reproductive fitness. And this is probably the reason why most of us actually are eating a little more than what we think we should, what we would want to eat if we were to live longer. The opposite of calorie restriction is obviously what we have currently in this country and across the Western world, which is an excess of calorie intake, which by all manifestation represents a disease of a form of accelerated aging. People who have obesity show accelerated atherosclerosis, type 2 diabetes, osteoporosis, neurodegeneration. So all of the diseases that we recognize as being associated with obesity and aging actually converge. So at this point, I would love, so I've just showed you data that suggests that both environmental lifestyle factors, but also genetic factors play a role in longevity. And I would love to pause to ask you to answer this question. What do you think for humans? Obviously, I said the genetic aspects are critical because they allow us to do experiments in a laboratory setting. But what many people are interested in today is what can I do about my longevity? Most of us are not going to be able to change our genes. So what do you think is the relative importance of lifestyle versus your genes that you have inherited from your parents in terms of your longevity? And I gave you a whole series of choices where lifestyle goes from 10% to 90%. So if you have made your mental note of what you think is the answer, I will show you what the answer is, because it is actually 90% lifestyle versus 10% gene. Meaning that, of course, genes are important in terms of regulating lifespan, but their relative role in your personal longevity is small. And the reason I'm showing this data is I think it is an incredibly positive message for all of us. It means that whatever fate your parents had does not necessarily apply to you. It also means that by adopting interventions or lifestyle factors that will mitigate the aging process, we are all in control finally of how the quality of our older age. And the Buck Institute, where I work, is actually really focused on understanding what these lifestyle factors are. And I will be devoting a part of my talk at the end to this very question. So what have I told you so far? First, aging is malleable, a lot more than we used to think. And in the next few minutes, I want to highlight some knobs, I call them knobs in lay term, that regulate aging. So these are molecular pathways that are able to control our aging. And these are obviously genes and the environment and the interface between the two. So just want to tell you a word about the Buck Institute where I work. We were founded 20 years ago as the first freestanding research institute in the world focused solely on the biology of aging. So we've been in business for 20 years. Today we have 24 research groups, 280 employees from over 30 different countries. We function as most research institutions that you are familiar with postdocs and students and technicians. We are not for profit. As I mentioned, we opened in 1999. We live in a beautiful IMP designed 250,000 square foot campus, about 30 minutes north of San Francisco. Our current budget is $53 million, 50% of which is actually supported by the NIH, in large part from the National Institute of Aging. And the rest is equally divided between corporate agreements that we have with pharmaceutical and biotech companies, 17% philanthropy, and 17% from an endowment from the Buck family. So our scientists have been busy, and I certainly do not mean to imply that we are the only one working in this field. This is a very rapidly growing field, actually one of the most exciting in biology today. And we have been working trying to understand these pathways that control aging. On this slide, I'm showing you a summary of much work that has been conducted, trying to understand what is the link between calorie restriction and a long life. And so, as I mentioned earlier, much of the work, the initial work has been conducted in C. elegans. The reason is the rapidity with which one can conduct a lifespan, as I mentioned, 20 days. If you move to Drosophila, it takes about 3 months. In mammals, it takes about 3 years. So by using the C. elegans, we were able to dramatically accelerate our work. Now, what has emerged out of this work is actually pretty remarkable because it turns out that the pathways that control aging, and I'm showing you here just the pathways that are responsive to calorie restriction, actually remarkably conserved between C. elegans, between Drosophila, the mouse, and actually in humans as well. A really good example is the TOR pathway shown here. TOR means target of rapamycin because this protein is actually can be inhibited by a drug called rapamycin. And remarkably enough, rapamycin itself exhibits some of the strongest anti-aging effects, both in Drosophila, in C. elegans, and in mammals. There are other players that were identified, so really showing the universality of these pathways and their ability to regulate aging. But TOR is not the only one. The insulin signaling pathway is shown here in blue. This is the DAF2 protein mutant that I showed you earlier that was regulating aging in C. elegans, is part of the insulin IGF-1 signaling pathway. The sirtuins I will be discussing in a little more detail because this is the area in which my laboratory has been working. So using this type of approach, we've been able to identify not only nodes and players that regulate aging, but also drugs that target these pathways. And I'll be discussing in a few minutes the relative roles of again, the environment, but also drugs in targeting aging in the future. So what my laboratory has been focused on is really understanding the link between calorie restriction and health span. And we have proposed a working model which says that calorie restriction as an intervention has profound effect on a number of intermediary metabolites that are shown here. So these metabolites play key roles. NAD is a key energy converter in the cells. Acetyl-CoA also is an intermediate form of energy. S-adenosyl methionine is the major methyl donor. Alpha-ketoglutarate is a key metabolite in the Krebs cycle. And beta-hydroxybutyrate is a key metabolite generated during a process called ketosis. And this is a topic on which we work. We've been able to show that every which of these metabolites, which are used as forms of energy, also speak to a number of key proteins that we call master regulators. For example, beta-hydroxybutyrate regulates the activity of HDACs. The NAD here regulates the activity of sirtuins. Acetyl-CoA regulates the activity of histone acetyltransferases. So these master regulators in turn regulate a whole series of proteins which mediate the effect on health span and lifespan. So let me give you an example also where this type of work has led to. So we know that calorie restriction induces a prolongation in lifespan. So there's been a lot of interest in identifying what we call calorie restriction mimetics. So these are actually small molecules, drugs, that fool the body in terms of making it believe that it is under calorie restriction. And what we've seen with these calorie restriction mimetics is that they induce a response which is similar to the absence of food. So obviously, calorie restriction is something that is actually quite difficult to live. There are people who have placed themselves under calorie restriction. It's been said by some colleagues that a calorie restriction might not make you live forever, but it will certainly feel this way. So for this very reason, we think that calorie restriction is not likely to be a practical intervention that we could recommend for most people. Although I would argue that all of us should watch our portion size, which is another way to mediate some degree of calorie restriction. So there's been a lot of interest in understanding what drugs actually mimic this process. And what I'm showing you here is a very busy slide, but I will not have a chance to go into the details. What I want to show you is many of the proteins that I've talked about, these sirtuins, for example, the histone acetyltransferases, mTOR, acetyl-CoA, these are all nodes that allow us to identify small molecules, and each of the boxes here highlight small molecules that are currently being explored for their potential as calorie restriction mimetics. I will highlight two in particular. Metformin, this is an anti-diabetic drug, also called glucophage. It turns out that patients, diabetics who are on metformin, we know today live longer than non-diabetics. And so we've also seen some evidence that metformin really induces a calorie restriction response in humans, and appears to be protective against Alzheimer's disease, against cancer, against type 2 diabetes, and against heart attacks. So these are large epidemiological studies that support a role of metformin perhaps as the first anti-aging human drug, which is right now being prescribed for diabetes, but might have profound anti-aging effect. A clinical trial to address the prospective clinical trial is currently being planned by Nir Barzilai and his colleagues at Albert Einstein, in which they will take non-diabetics and randomize them to metformin or control. These patients will then be followed for a period of 5 to 7 years, and they will measure the incidence of some of the chronic disease of aging that I've highlighted. I will be discussing a bit the role of NAD precursors in a few minutes. I just want to highlight the fact that there is a wealth of new drugs and new targets that are being currently explored by this field. And I can predict that within the next 10 years, we probably will have in the clinic the first anti-aging medicine approved directly for that purpose. So there's a lot of excitement in the field, and I want to highlight also towards the end of my presentation why we think these medicines are going to be really important. Again, here, the primary target is not so much to make people live longer, it is to make them live better and to suppress the development of these diseases. And I will be discussing this further. So I want to speak a little bit about the sirtuins. Larry has asked me to actually dig a little deeper and to, for those of you who have a bit more background in biology, I hope that this will satisfy your hunger for a little deeper information. For the rest of you, I hope that I will be able to make these discussion understandable. So my laboratory is focused on this family of protein called the sirtuins, and as I mentioned, they are really key in the aging process. So what are they? They are actually histone deacetylases. They don't only remove the acetyl group shown here on histone proteins, but also on non-histone proteins. Think about this acetyl group as a switch that allows this histone protein to behave in an on or off manner. These switches can be deposited on many other histone and non-histone proteins. And the sirtuins, by removing the acetyl group, is able to actually change the function of the protein and therefore to modulate aging. Now, one property of the sirtuins is remarkable is that they're using NAD as a co-substrate. And in this reaction, the NAD is cleaved between the nicotinamide and the ADP ribose, generating acetyl ADP ribose, which accepts the acetyl, and then nicotinamide. Now, there's extensive evidence that SIR2, which is a yeast protein, is part of the calorie restriction response. If one erases this, makes a deleterious mutation in this protein, in this gene, C. elegans, Drosophila, and mammals actually lose their ability to respond to calorie restriction by an increase in lifespan. My laboratory a number of years ago cloned the mammalian sirtuins, and we identified 7 proteins that are shown here. They all have a conserved catalytic domain, which is shown here in blue, which exerts this enzymatic activity. But they also had variable N and C terminus, which confer to each of these proteins different activities, different subcellular localization, and different specificities. So we've been quite busy studying them for the last 20 years. We have focused on 31, 32, 33, 4, and 5, and we have not worked on 6 and 7. But others have. There's quite a lot emerging literature in these proteins. One of the remarkable thing that we have found is that both 31 and 33, on which we have spent most of our time, are themselves regulators of a number of proteins that are known to be key regulators of unique pathways that modulate aging. I'll give you an example. 31 regulates PGC1 alpha, which is the master regulator of mitochondrial biogenesis, regulates FOXO1 and 3. FOXO3 is one of the genes for which we have polymorphism that are associated with a long life in humans. NF-kappa B is the master regulator of inflammation. One of the characteristics of aging is a global increase in chronic inflammation. PPAR alpha is a global regulator of lipid and glucose metabolism. SIRT3 is a mitochondrial sirtuins, and he also regulates a number of key aspects that have direct bearing onto aging, such as regulating oxidative stress, which is imposed by the generation of these radical oxygen species, but also regulating mitochondrial biogenesis. Superoxide dismutase here is a key enzyme also in oxidative stress. Now, because of this work, so this was the last 10 years, from 2000 to 2012, there was a lot of excitement that the sirtuins might actually be incredible players against aging. Companies were started, but this was rapidly followed by a lot of disappointment. That is, a gain of function, activating the sirtuins, did not lead to an increase in lifespan. And so it took a little while for all of us in the field, to realize that the reason that the sirtuins did not work to increase lifespan was that as we age, the critical cofactor for the sirtuins, called NAD, actually decreases. And so this has led many of us who were working on sirtuin to shift our focus on trying to understand what is the reason for which NAD levels decrease, and can we restore these NAD levels as we age. The implication of this is that by restoring NAD level, we will reactivate the sirtuins, who will exert their beneficial anti-aging activities. So actually 4 years ago, I proposed a hypothesis, which was that NAD is actually being overused by other NAD utilizing proteins. And there are three broad categories of protein that actually consume NAD. The sirtuins, I've just introduced you to their activity, but also PARP, poly-ADP ribose polymerase. These are key proteins involved in DNA damage, which we know increases during aging. And in addition, two proteins that we are currently studying, called CD38 and CD157, who are NAD hydrolase, so they consume NAD, and they play a key role in the immune system. So when these proteins, the sirtuins, the PARP, and CD38 utilize NAD, they degrade it and they generate this product called nicotinamide, which needs to be recycled into NAD. And so there's been a lot of interest in trying to understand what is the relative role of a defect in recycling versus overactivity of the PARPs and the CD38, so that the NAD levels decrease, and therefore the sirtuins do not have what they need to exert their activity. And I just want to point to the fact that this led to a lot of interventions. As I mentioned, resveratrol and the stacks were drugs that we know people tried to use to restore sirtuin activity. That didn't work. Some groups have actually tried to target the PARPs to generate inhibitors. Interestingly, if you inhibit PARPs in a little worm, you can actually increase lifespan and you restore NAD levels. However, the PARPs are also, as I mentioned, important for DNA damage repair. And so in long-lived organisms like us and others, the PARPs are really critical to maintain genomic integrity. There's been some focus also on trying to understand why does NAMPT, this is one of the key enzymes in the salvage pathway, why does it decrease during aging? There's also a lot of interest, and some of you might be aware of this, in supplements that might actually be able to restore NAD. Currently on the market, there are two supplements. One is called nicotinamide riboside, or NR. The other one is called nicotinamide mononucleotide. There are a number of companies that sell these. They have not been proven to, they've been proven to restore partially NAD levels during aging, but there are still a lot of questions in terms of their efficacy and their safety. Finally, there's the identification of CD38 and CD157 as other enzymes that actually degrade NAD during aging, has highlighted the possibility of new targets, and a number of companies are actually currently exploring this possibility by looking for novel inhibitors of these proteins. So I think the prediction is that in the next few years, some of this work is going to be panning out, and we will be able to restore NAD levels during aging. The prediction is that this will have a profound effect on lifespan and health span. So I want to go back now and from this deep dive into the biology to discuss what are the implications of this work for human health. Obviously, all of us, one of the great pleasure that I've had in working in this field is I do not have to justify to anyone why we are working on this. This is something that implies all of us. It is a long life quest for humanity to actually improve its quality of life as we age. And so I just want to remind you that aging is the strongest risk factor for age-related diseases. A good example for this and what this means, I've shown you now in the last half hour that that aging is malleable. When I went through medical school, we were told that aging was indeed a risk factor for diseases, but it was also considered something like your gender, something that you could not change. So if you could not change it, it was therefore of little interest. What we've learned in the last 20 years is that aging is malleable and that we can change it. The implication of this is that if we can change aging, we can change the occurrence or the rate at which we are developing these diseases. And this is what really is motivating the field right now. I want to show you also, want to share with you the importance of aging. If you think about one of the known, if I ask you to think about risk factors for heart disease, most of you will be able to recite many of them, and I'll go through them: obesity, high blood pressure, high cholesterol, type 2 diabetes, hypertension, inactivity, and so on. Many people today in the US are actually on the statins, which is a medicine that lowers your cholesterol. And the reason they are on these statins is because these medicine has been shown to lower your risk of a heart attack. Now, what's remarkable, if you think about a high cholesterol indeed increases your risk of a heart attack, but as a risk factor, it is 7 times less important than your age. So the age is actually an incredibly strong factor for all of these diseases. This is best illustrated by the logarithmic nature of this curve. So what is and how is medicine today functioning in the 20th century? I've illustrated this with this drawing, which shows medicine comprised of a number of silos. If you have heart disease, if you're, God forbid, you're 65 years old and you have a heart attack, and you're going to go see a cardiologist. The research that is being conducted on heart disease is by the National Heart Institute. So our medicine is organized in silos that are actually based on an anatomical organization of the human body, which dates back to the 16th century. This view of biology needs to be revised, and I'll show you why. So imagine, let's continue to imagine the story of our 65-year-old patient who has had a heart attack, has survived, and now sees a cardiologist, and now survives but continues to age. It is very likely that within the next 5 years, this patient will actually now develop a cancer. Now, we're making incredible progress in cancer treatment, as you have heard. And to be treated, you will go see a special cancer doctor. There's a National Cancer Institute. Again, we organized our institute in terms of diseases or organs. Now, it is remarkable to actually think about how much would lifespan increase if we were to cure cancer tomorrow. And the number is actually 3 years. So this sounds a discrepancy with what we actually are expecting. And the reason is that if you survive your cancer, you are now at increased, you're going to live another 3 years, and now you're going to be at increased risk for Alzheimer's or Parkinson. And so goes the list. So right now, our medicine and the way it is practiced today, we've called it humorously as whack-a-mole medicine. It is attacking these diseases as if they were independent manifestation. And I certainly do not want to diminish the incredible work that's been done by my colleague and myself in the past. I'm a physician, so I'm part of the system. But the work that we are doing in aging actually calls for a new and a renewed understanding of what these diseases truly represent. So what we actually are proposing is really change medicine from what it is today. We call it healthcare, but actually it is not healthcare. It is sick care. It is organ-based. It is highly reactive. There are some attempts at preventative medicine, but they work to some degree, but this is not the heavy focus. Most of our actually dollars that are spent are are spent on disease management. And most of the time, our interventions are universal. Many of you have heard about precision medicine, molecular medicine. These are all ways that highlight the fact that as individuals we're quite different, and that many of the interventions in the future will be highly tailored depending on our genetic background and risk factors. So we hope to work at changing this model into a true healthcare system, which will be system-based. When I say system-based, is instead of studying organs, I think we have to study processes. And the pathway that I've showed you that regulate aging, these pathways not only regulate aging, they also regulate health span. So they regulate both lifespan and health span. And in most cases, these mutations that I've showed you not only make animals live longer, but they also lead to something that we call the compression of morbidity. That is, these animals live longer, but then also their fraction of their lives that they live sick is shorter. So this would allow us to change a medical system which is reactive into a proactive medicine, preventative medicine, and highly personalized. So we are quite excited by this vision. We are in the process of building what we call Buck Healthspan 2020, whose goal is to increase health span and lifespan by 20 years in the next 20 years. I think this is a very conservative estimate. I predict that based on what we know today, we can optimize lifespan in most individuals to live to 100. Now, this initiative is focused on a number of areas. Biomarkers of aging is the ability to measure. I mentioned earlier in my presentation this idea of identifying biological age. Think about the two trucks. And the area of biomarkers, think about what it means to measure your cholesterol level. There are now a number of markers that have emerged that allow us to really precisely identify even in younger individual, their trajectory in terms of aging. Think about also the way we practice medicine. As I mentioned, we wait typically for people to get sick. You can be a very healthy 65-year-old man and the next day have a heart attack. Obviously, we want to do better. We want to be able to identify people in their 20s and 30s and 40s to really better predict who is at risk for these diseases. And these biomarkers of aging are going to be doing this. There's a proliferation of work in this area. It's quite exciting. We also want to work primarily on lifestyle intervention, food as medicine, exercise, sleep, drugs and supplements, and very far in the future, there's a potential also of using genetics. So we're quite excited by this new initiative, and we are building these programs one at a time. And I'll be discussing very briefly at the end, and I hope we'll have some time for questions to really address what do we know today that actually optimizes your health and your health span. So, and that's a good segue for me to move into my next question is how and how much will we improve health span and longevity? I would venture to say that it probably will not be this way, although I love this picture because it is representative of one aspect that is really key. I've mentioned to you that 90% of your lifespan is determined by lifestyle. That is true for most of us, except for those of you who have centenarians in your family. It turns out that many of the centenarians today actually have very strong genetic effect that make them live longer. And they've been studied extensively by a number of my colleagues, including Nir Barzilai, who's built a very large cohort of centenarians. And this example is actually a true example of a patient who is still smoking at 100. So if you have someone in your family as a first degree relative who has lived above 100, it's extremely likely that you will live, you will actually have the same fate. Now, the question that most of us actually are interested in, what should the rest of us do to increase our longevity? And one of my favorite quotes in the field is actually not by a scientist, but by someone who had incredible wisdom, Jack LaLanne, who said, exercise is king, nutrition is queen. Put them together and you've got a kingdom. And I think he could not be more correct. He lived to 98. even though his father actually had died in his 50s from a heart attack. This early death is what motivated him to adopt this lifestyle, and it did him very good, very well. He lived actually to 98. So what should you and I do if we don't have these great genes that will make us live longer? I have a slide here that I would be happy to bring back during the questions if you are interested, because obviously a lot of us want to have drugs that target aging. These drugs will come. There are some big questions in terms of who will receive them, at what age will you take them. As a physician, I'm concerned about the idea of giving a medicine to someone who's healthy in their 50s or 40s with the hope of making them live longer. So it's very likely that these drugs initially will be restricted to people who are at increased risk. However, they will be there. I can tell you that metformin sales right now are up 20% in the last 4 years, simply based on the data that shows that patients on metformin are living longer. I'm quite often asked, should I take metformin? And I think this is a very difficult question that I would ask you to, that I would recommend that you discuss with your physician. Nutrition is, so what can we do beyond drugs? I think this is a really incredibly interesting topic, but one that is really emerging. And if you ask me what should I do in terms of drugs, I would say, I do not know. We do not have the answers. We will in the next 10 to 20 years, but right now we do not know. So what we do know is a series of other interventions that you can do that will increase your lifespan. This include nutrition. There's a lot of focus on in terms of nutrition, in terms of what diet should I follow? Should I eat a low-fat diet versus a low-protein diet versus a low-carb diet? I think this is a wrong way to look at this. There's a lot of emerging data that it's not so much what you eat, it's more when and how you eat. There's growing evidence that fasting, remember fasting is in some way a calorie restriction mimetic. It is one way in which we can trigger the same protective response that calorie restriction will do simply by fasting. A lot of work, a lot of research ongoing in this area. The same in terms of exercise. It's clear that regular exercise is the ultimate anti-aging medicine today. A lot of people will tell you, well, I don't like to exercise. I can tell you that walking 25 minutes a day has profound effect in terms of longevity and health span. And so I would argue that every single person should walk 20 to 25 minutes a day if you want to have a healthy old age. There's a lot of work ongoing there, also understanding how different forms of exercises such as endurance, strength, stability, flexibility interface with all of the pathways that we know regulate aging. Now, obviously, other factors can be critical, such as your environment, the quality of your air and your water, oral hygiene, your microbiome. Sleep is one big area right now. There's growing evidence that as a population, we are chronically sleep deprived, and that sleep deprivation has profound effect on many aspects of our aging phenotype. Now, I love to bring up social, friends, and service. So having friends and having the feeling that you are of importance to society are actually the two strongest predictors of your lifespan. This is the result of a large prospective study that studied a number of markers over a 10-year period. It turned out that the quality of your social life and your sense of purpose in life were the two strongest predictor of a long lifespan. Now, the reason I bring this up is because it is easy to start worrying about everything, about your exercise and your nutrition, and to adopt lifestyles that are actually not compatible with a social life because your diet is too restrictive or because you are actually exercising all the time. So I always tell people, at the end of the day, having a great social life is one of the strongest predictor. And we know loneliness is actually one of the strongest predictor of an early death. And this is something that is actually of high concern right now, given the loneliness imposed on many of our older citizens due to lockdown and COVID-19. And so it is really a tough situation right now, and I think one that I think we all have to consider. Obviously, vaccines are coming back to the front. Everyone really understands the importance of vaccines in the middle of this epidemics, but there are it is actually amazing today to realize the fraction of people who are not getting their annual flu vaccine, and we know that 30,000 people every year die from the flu. The same thing for pneumonia, shingles, and etc. Obviously, traditional medicine is not completely failed. It actually is what has gotten us to where we are today in terms of, and preventative medicine is offering a number of really key issues such as colonoscopy, pap smear, breast examination, blood pressure, blood sugar, cholesterol, and so on. And finally, there is some room for blood work and supplements. A large fraction of the population is defective, has deficient vitamin D, many vitamin B12 as well. I support the idea that if you measure these and you find that you are deficient, there's very little to be lost by supplementing. So I want to finally answer the question, will we live to be 150 years old? I realize I'm almost at the end. I have a couple of minutes, so I will go quickly through this. I want to show you actually how we got to where we are today. So in 1850, life expectancy was 42. And remarkably, lifespan has been increasing monotonously for the last 150 years. So today in the US, it's 78. It's actually been increasing by 2 years every decade. So if we take this curve and we project in 2100, everyone should live on average to 98. I believe that we can make this curve go a little faster, but no one can predict where this is going to go in the future. As I mentioned, this looks like an incredible achievement of modern medicine, but it is counterbalanced by what is the blue area, which is the years of chronic disease that many of us have to endure if we age. It turns out that your health span is not increasing at the same rate as your lifespan. That means that as the population continues to age, the fraction of people who are going to be sick with these chronic disease of aging is likely to continue to increase, which is going to be a significant problem. So our vision of what the future is, and this is what the Buck Institute is working on, is really increasing your health span. It's not so much focused on lifespan, but really focusing on increasing the quality of life. Now, what makes me think that we can do this is we know the centenarians not only live longer, but they also live disease-free. This is the disease-free survival of controls and centenarians. So they live longer, but also healthier, longer. And this is best illustrated on this slide. You and me have both a health span and a lifespan. We live on average to 80, 78 to 80, and we developed our first chronic disease of aging around 68. So we spend about 15%, 12 years of our life afflicted by these conditions. Centenarians typically live to 100 on average, and they spend about 5% of their lifespan afflicted by these diseases. So our goal is essentially to transform everyone into what the life expectancy and health in terms of health span and lifespan that centenarians live. So I am 1 minute after my time. I want to finish by showing you a picture which actually keeps inspiring me. This is a quote by Carl Reiner and his friends who are all above 90. Dick Van Dyke, Mel Brooks, and Norman Lear. Sadly, Carl Reiner actually just passed away. And he said, every morning before having breakfast, I pick up my newspaper, get the obituary section and see if I'm listed. If I'm not, I'll have my breakfast. I think these guys are all, actually until he passed away, are all very active, still working, and enjoy the bonds of friendship. And I think this is the type of life that we can all aspire to. So I want to thank you for your attention. I invite you to contact me if you're excited about this work and want to visit the Buck Institute in the future. And I very much look forward to your questions and to our interaction.
And thank you very much, Eric, for a very entertaining talk and for diving deep in parts to to the molecular biology that underlies our understanding of some of these aging processes and interventions. So we have time for Q&A, which we often take a lot of questions. Zoom participants, raise your Zoom hand and we'll unmute you in turn to ask a question. If you're a Zoom participant, you don't have a microphone or you don't want to ask directly, you can submit questions in the Zoom Q&A panel. And YouTube viewers, text your questions in the YouTube chat box. All text questions will be read aloud for everybody to hear. And let's begin with a question from Carl Merrill.
Yes, so thank you very much. Eric, I really enjoyed your lecture. I have to admit that I was actually interviewed to be one of the first directors of that institute years and years ago. And I've continued to work to some extent on aging. There are a couple of areas that I was interested in that you didn't touch on, and I wondered if you'd comment on. One was parabiotics. I wondered if the Institute's doing anything on that. And let's see, the other question I had is, it's one thing to make a discovery about something, and it's another thing to get it out to where it can be used by the population. And I retired from the NIH back in 2012, but I'm actually active now trying to make a vaccine for the COVID-19. However, I did work on Alzheimer's disease. And in fact, the sponsor of our lecture tonight, Tim Thomas, helped out with that. And one of the things we discovered was that there's a decrease in protein synthesis in areas affected by 40%. And we actually narrowed it down to elongation factor 2, which happens to be the target of diphtheria toxin. So the question was, what happens if you just get a booster shot when you're 50 or so? Will that decrease it? And Tim actually helped me with the analysis of data from all different cities. And it looks like people who did get booster shots did have a decrease in Alzheimer's. I tried to get people in clinical medicine interested, but really didn't succeed at all. And I wondered how your institute tries to bridge that gap from your fundamental discovery to the point where you can really test it in a population.
Okay, very good. So let me answer the first question, the one on parabiosis. So for the part of the audience who does not know what parabiosis is, it is a discovery that was conducted by Tom Rando at Stanford. What Tom did was to essentially sew two mice together, and was able to show that, so these two mice now had an old mice and a young mice together, shared their circulatory system, and was able to show that when you do this, these two mice now have a single circulatory system. It turned out that the old mice got younger and the young mice got older, implicating the existence of factors circulating in the blood that accelerate aging and slow down aging. So this was followed up by demonstration that the plasma, if one takes the plasma of a young mouse and injects it into an old mice, you can actually see a reversal of aging. And actually today, there are clinical trials from a company called Alkahest that is taking the plasma of young people and giving it to older individuals with early cognitive impairment or early Alzheimer's. And the preliminary results that they have reported showed a beneficial effect. So this obviously will lead to the identification of blood factors, single factors, not the whole plasma, that one would hope could exert anti-aging effect. So this is a very exciting area of investigation. Many of our scientists actually are using these types of intervention in their respective model systems. So very exciting area. The second question that you asked me was, how does the buck actually transfer its basic research into the clinic? And I think this is, I love this question because this is why I took this job. I'm an MD by training, and I've always done research with a purpose. With a purpose beyond just the principle of discovery. And the purpose for me was to improve human health. I felt that the reason I left UCSF and the Gladstone in 2016 to take this job at the Buck was to really come and take all of this basic knowledge and help translating it. And what I highlighted as our largest initiative right now, which is this Buck Healthspan 2020, is really our vision to take all of the basic research and bring it to the clinic. And we're going to be doing this in three different ways. One, we are enhancing our translational research, which is research conducted on human tissue, including stem cell biology or IPS for those of you who are in the field, working more in collaboration with clinicians, building a clinical center and a clinical research center at the Buck. And finally, last but not least, is we have a very active business development program. I think academia is great at generating ideas, but at some point someone has to take these ideas and translate them into the marketplace. And so the Buck has been very actively spinning companies. We've spun 5 companies in the last 3 years, and we have a lot in development. Now it turns out that there is a lot of appetite right now in the investment world for this type of research. And I think we think that a combination of a strong basic research, a focus on applicability to humans, and a translational effort in spinning businesses is going to be the magic sauce that's going to allow us to not only do incredibly interesting work, but also to change humanity and to change healthcare.
Thank you. That's exciting.
Thank you.
Thanks, Carl. We have a question from Frederica. Frederica?
Hi, and I also enjoyed very much your presentation. Actually, I had 2 questions. You mentioned the artificial light. What did you mean as part of the sleep kind of conditions? And the other is continuing to work and not retiring as early contributing to the health span?
They're 2 very good questions. So what I mean artificial light is I think we have to blame Edison by bringing to us the ability to continue being exposed to bright light throughout the evening. Many of us have disrupted sleep. And there's a growing recognition that sleep hygiene is actually incredibly important in terms of longevity. Now, there are a number of approaches that are being taken. As you know, there are unique receptors for blue light, so melanopsin receptor, which apparently plays a key role in this. So there are approaches consisting of trying to block this blue light. You might have seen some people wearing orange glasses. There are applications on your telephone and on your screens that might actually also contribute to this. But this is just part of a very large picture. I think there's an increasing recognition that, and this is best exemplified by shift workers who actually suffer from an accelerated chronic disease of aging. When you look at people who are constantly shifting their schedules, they actually show a phenotype that one could describe as accelerated aging, increased risk of heart attack, metabolic disorders, Alzheimer's disease, and so on. So I think this is one area in which I personally try to make a lot of effort in terms of having a good sleep hygiene, sleeping enough, and having a regular sleeping times. I'm sorry, I blanked out of your second question.
Continuing to work rather than retiring.
Yes, I believe, the data shows that there's a clear peak of mortality on the year after you retire, which actually is really consistent with what the epidemiology says, is that if you lose a sense of purpose or a sense of relevance, people are essentially at increased risk for all of these diseases. Now, this doesn't mean that I'm against retirement, because I think it's not so much the act of retirement, it's the loss of purpose and the loss of relevance to society that actually serves people. I still believe in a society where people can retire But the key is to try to find something that allows you to keep that sense of importance to your family, to your relatives, and to society in general.
Thank you.
Okay, we have a question from Joel Wilson, another PSW member and a physicist.
Yes, hi, Doctor. I really enjoyed your presentation. I noticed you mentioned during the presentation about inflammatory responses. I'm wondering if there's been a mapping about that signature of inflammatory causes, like excess weight is really kind of an inflammatory condition. Foods that are processed and might have a little bit of sugar, and sugar can generate an inflammatory response, and how shifting away from that might extend healthy lifespan. Has there been any kind of mapping on that, sir?
Yes, there's a lot of work. It's a very good question. A lot of work in this area. As you know, there's a concept of inflammaging, which is the fact that if you look at the blood, so first there's two forms of inflammation. I mean, there's acute inflammation. This is the response of your body. If you get burned, for example, you will see redness, you will see pain. So these are manifestations of acute inflammation. There's also evidence that as we age and as the damage that is associated with aging increases, one sees a sterile form of inflammation, not caused by an infection, where you have many of the same markers that are circulating. And for example, we have some data in the lab that shows that this inflammation actually lead to NAD depletion. So this is another cause of NAD depletion. And CD38, for example, is a very strong marker of inflammation. So the question is, so the thinking today is the inflammation represents a marker from the damage. As you age, you accumulate the damage, and this damage is recognized by the body as an intrusion. And therefore the body mounts this inflammatory response. Now there's also what you pointed to is this idea that some of the inflammation can be caused by the diet. There's a lot of discussion in the lay press about inflammatory diet. I would say that data is a little softer. It's not totally clear. I've been very interested in this, in exploring this further to see really if there's such a thing as an anti-inflammatory diet. Clearly, we know that a high-fat diet with saturated animal fat is really associated with chronic inflammation. Beyond this, it's a little less clear, but there are also clear links, for example, between the insulin signaling pathway and inflammation. So one of the thinking is that many of the manifestations of aging actually all converge on chronic inflammation. And so we think that this chronic inflammation could be one of the targets in the future that we will go after to prevent these chronic disease of aging. Many of them are also driven by inflammation. So very good question.
Thank you, sir.
I'm going to take a question from the web, and then we'll try and get back to Tim Thomas. So the first one is a kind of prickly question from somebody who apparently has been following this for a while and wants to know, we have heard the promise of longer lives, more than 10 years now. What is the actual likelihood that we will get the therapies in the next 5 to 10 years that science keeps on promising?
Yes. So, as I mentioned, there are two aspects to this question. One is perhaps this member is asking about the drugs. The drugs are in clinical trials, and I think these clinical trials, one of them failed recently. Developing drugs is a long, arduous, expensive process, which is riddled with failures, not unique to the aging field. So I think we can expect, I mean, typically 70% failure for 30% success. So right now there are several ongoing clinical trials that are dealing with different aspects of the aging process. And I predicted earlier that within 10 years, there will be a drug that targets some unique aspects of aging. Now, the 10 years that you want to gain in lifespan, that knowledge is here today. I predicted, and I think if you look at different areas, the best way to, so why do I know that actually if you were to optimize your lifestyle, I would predict, depending on how you live, that you could gain another 10 to 20 years of life expectancy and health expectancy. Now, why do I know this? Is that I have the good luck of living in Marin County, which has the longest life expectancy in the country. Now, life expectancy in Marin right now is 87 in comparison to 78 for the rest of the country, which we know is likely a reflection of lifestyle, education, socioeconomic status. Now, one thing I can tell you is, because I live in Marin, is that not everybody here is optimized. Not everybody exercises. Not everybody eats healthy. There's still a fraction of the people who smoke. And so that allows me to say that, if you look at, in comparison to the rest of the country, if we were to optimize a whole county as Marin, my prediction is that we could go from 78 to 98 lifespan. And I think the vision of this healthspan center is to put this into practice and actually demonstrate it.
We'll go on to Michael Helton.
Yeah, I'm sorry, Tim, we could not understand you.
What part does the immune system play in aging? Can a bolstered immune system increase health span and lifespan? I think that's a bit, but go ahead.
That's a great question, and one that is actually highly relevant to the COVID-19 epidemic. It turns out that we have two immune systems. One is called the innate immune system. This is the immediate response immune system. That immune system is overactive. This is what we call the inflammation. Inflammation is a reaction from the immune system. So, and this overactivity of the innate immune system is what's causing actually the problem that people with COVID-19 who are older suffer from what you might have heard as a cytokine storm. So the cytokines are mediators of inflammation. Now, the second part of the immune system is the adaptive immune system, which is the one that you trigger when you vaccinate. And that one is not overactive, it is less active in older individual, which means that they are unable to respond to the virus. This is the form of immune system that you trigger by a vaccine. And this has really profound implication for COVID-19, because we know if you are above 70, the likelihood that you will respond to a vaccine is probably around 30%. And this is true for the flu or for any other vaccine. So how will these vaccines work? They probably will work via a herd immunity effect. And so we are actually working on both aspects of the immunity. There are lots of changes that happen in the immune system. The key thing about the immune system is that it has to be exactly in balance. If you remember the slide that I showed you about eating, this bell curve, it's the same about the immune system. If you have an overactive immune system, you can get inflammation, or you can get autoimmunity. And we know as you age, there's an increase in autoimmunity. If you have not enough immune system, you have actually immunodeficiency. Both are bad. And so the idea is to really create a situation right in between, where your immune system is active when it needs to be and inactive when it needs to be as well. Now, what we know is that all of the pathways that I've discussed, metformin and all of these pathways that regulate aging, are all present in immune cells. And therefore, if you have a lifestyle that is healthy, your immune system is going to be healthy as well.
So I'll read the question from Tim Thomas. He asks, what do you think of the prospects for genetic manipulation of mammals for the purpose of extending lifespan? He's thinking especially about dogs, since people are willing to invest in dogs, love their dogs, and their dogs tend to die long before they do.
Yes. very exciting. So this is actually being done. There are a number of companies that exist that have been doing exactly this, which is introducing genetic type of intervention in dogs. It's obviously something that we can do in dogs, but we cannot do in humans, at least not now. Trying to see if you can revert aging and prolong their lives. There are also clinical trials for rapamycin, for example, for dogs. There are very large clinical trials coming out of University of Washington. So there's, I think this, you know, this is a field of investigation which is incredibly interesting. Obviously, we can do many things in animals that we cannot do in ourselves, but it might be a harbinger of what's to come for us in the future. Obviously, in humans, gene therapy is restricted to cancer and to some very rare congenital diseases. But as we learn how to manipulate genes much better, I think I don't think there's nothing unethical about the idea. It's just that we're certainly not ready to do this in humans.
Interesting. We have a question from a new member, Omar Chalan. You can ask your question, Omar.
Hello, President Milstein. First of all, thank you so much for accepting me to the society. I'm so delighted to be a member. And my question to Dr. Eric is, I have been reading the book titled Together for Dr. Vivek Murthy, the former General Surgeon of the United States. And he mentioned a very interesting, some results for a very interesting study for Dr. Julian. She's a psychologist from Brigham Young University. She found in her study that the impact of lacking social connection on reducing lifespan is equal to the risk of smoking 15 cigarettes a day, and it's greater than the risk associated with obesity, excess alcohol consumption, and lack of exercise. Simply put, Julian had found that weak social connections can be a significant danger to our health. So my question, it's not a question, I just was interested to hear your comment on this striking results.
No,
Thank you.
I mentioned it. Thank you for the question. It's I mentioned this during the presentation. This is there was a large prospective study where I believe 7,000 men and women were assessed at baseline at age 50 and then followed for close to 10 years, during which some of them became sick and some of them died. When looking back at what were their defining characteristics at age 50 of the people who died the earliest, lack of social connection was one of the two top factors. And the other one, I think, as I remember, this was lack of first degree. It could be either family, but it did not have to be family. It could be friends. It could be someone with whom you're sharing a very intimate connection. And the second one, I believe, was the lack of a sense of purpose or social relevance. And I think this really speaks to exactly the same thing that you mentioned.
Thank you so much.
You're welcome.
Delighted to have you as a member. So thank you. We have another question from the web. Another PSW member, Will Angel, asks, how can we provide equal access to increase longevity? Recent changes in mortality in the US, at least, have been unequal along socioeconomic lines. Is long life only for those in the upper socioeconomic brackets?
This is probably the most important question we've heard tonight, because it turns out that the strongest predictor of your life expectancy is actually your zip code. Now, obviously, this work has no meaning if it is targeted to the rich and the affluent, and I think this is the same for all of medicine. So my vision is really that this work has to benefit everyone. Now, there's some concerns because some of the donors have been billionaires who have actually invested heavily in this field, and there's a part of me that has sympathy for the billionaires. If I were a billionaire, I also would want to live forever. But obviously, this is not why we're doing this work. One good analogy that some colleagues have used is the Tesla model. Tesla started as a company by building cars that were restricted to the affluent, but soon using this technology and this knowledge, they were able to build the lower Tesla, and now they're talking about a $20,000 car. So obviously, we need people to invest. We need investors in this field. The hope is to have medicine that will be available to everyone. The best example actually of this and the potential is metformin, which costs 28 cents per day to take today. So it's certainly not something that's going to be restricted in terms of a socioeconomic status. So I think obviously you're bringing up an important point: is how do we bring about to society more equality that will allow people to, it's very easy to start worrying about your nutrition and your sleep and your diet if you have the time, if you're working 3 jobs to meet the needs of your family. Obviously your health becomes secondary. So I think these are really important questions. As a scientist and as a physician, I cannot solve every society problem. I think I can continue working to providing answers, which then will have to be developed in collaboration with the physician, with the politicians, and as a society that we will have to make the decisions that actually promote health in our whole population.
We have a question from our treasurer, Brett Magarum. Brett?
I was just curious, what are there any negative side effects to the drugs that are in trial? Because obviously, if these drugs work, you're going to have a lot of people clamoring to get them immediately. And as we've kind of seen with COVID, what could be a potential cure usually winds up having negative side effects that people are not aware of in the beginning. Thank you.
Well, it's hard to speak of what the side effects of individual drugs are going to be. One thing that we clearly see is that that we can reasonably predict is that longevity and fertility tend to go in opposite direction. Right now, we have a population which is living much longer than before, with a significant decrease in fertility. It is one of the things we've seen in many of the interventions, mutations, and drugs, is that those that increase lifespan are also associated with the decrease in fertility. So this is something to consider. Otherwise, there will be side effects with drugs, and this is really, for me, one of the key things to resolve. These are side effects that have to be compatible with a high quality of life. Otherwise, it just doesn't make any sense.
So we have kind of a follow-up question from the web. It's more specific along the same lines, but asking for your comments on the safety risks associated with metformin and NMN supplementation.
About metformin first. Metformin has been prescribed in hundreds of millions of doses to diabetics across the world. Actually, you can buy metformin without a prescription in Spain. It is an over-the-counter medicine. In this regard, it is an extremely safe medicine with very low rates of complication. On the other hand, I always tell people it has side effects. For example, there's an effect on testosterone. So if you are working out and you're hoping to bulk up, so there might be a decrease in your ability to have big muscles. Same thing, decreased in sex hormones. So the situation. So, and I think this is really a very important thing for people to consider. There likely are no free lunch, and especially if we consider giving medicine to people who are healthy, we have to have an extremely high bar in terms of safety. I would argue that most people who exercise and eat a healthy diet or fast or do some of these interventions should probably not be on metformin unless they are at high risk. If someone in your family has suffered from Alzheimer's and a high rate of heart disease, this is something to consider. Now, with regards to NMN, this is really the other side of the aging field, which is these are sold as supplement and therefore have not been tested. They've been tested extensively in animal models, and having worked on NMN and NR, we see remarkable things in animal model systems. But no clinical trials, at least no large-scale clinical trials, have been conducted. I would predict that these are relatively safe. There are a number of companies who sell them. We haven't heard of any untoward effect, but if you take them, you should always be aware of the potential risk.
I have a question from PSW member Al Ehrlich. Are there an effectively unlimited number of stem cells in an individual mammal, and do they age along with the other cells of the body at about the same rate? And does the answer to these questions vary from organ to organ?
It's a very good question. Yes, stem cells age and they progressively lose their regenerative potential. So when I discuss this idea of repair and maintenance in many organs, it is in large part caused by the ability of these stem cells to restart from zero. We know from the work of many colleagues in the field, Emmanuel Passeguet and Tom Rando, among others, that there is evidence for aging of stem cells in the bone marrow, which is responsible for all of our blood cells, in muscles for Tom Rando. There's evidence for this in the liver as well. So I think it's quite, and there's a lot of interest in understanding what are the factors driving aging in these stem cells, and to alleviate, and whether some of the interventions that we're pursuing are actually alleviating these aging phenotypes. So this is an active area of investigation for the future. It also is, the stem cells have another particular relevance to the aging field, because there are two ways that we think about aging and intervention. One is to prevent aging from occurring. The second one is to repair what has been aged. And for example, there is a remarkable work ongoing at the Gladstone Institute by Shinya Yamanaka and colleagues who've discovered factors that allow us to reprogram somatic cells, for example, skin cells, into stem cells. And these stem cells then can, in the future, will be able to be used to regenerate tissues and to repair what has been damaged. I'm thinking of a heart attack. They have preliminary evidence in mice where you can actually restore cognition in mice who are suffering from early Alzheimer's by reimplanting stem cell derived neurons. So I think this is an area of investigation for the long-term future, but one that carries a lot of promise.
I want to ask a follow-up question, and then we'll go to Lloyd Mitchell, who's another PSW member and a molecular biologist. My question is, so when stem cells are propagated in vitro, one of the characteristics is they're virtually immortal. And it's thought in the body that they divide very slowly, but they are essentially immortal. So I guess my question is this: to what extent is the observed senescence of stem cells, do you think, is genetic to the stem cells, inherent to the stem cells, or a factor of changes in the compartment in which the stem cells are usually found?
So that you're raising a very good question. That's the whole concept of a niche. So it is true that a stem cell's definition is its ability to replicate in a way that is immortal. And there is a growing concept, which is the idea that these stem cells do not, at least in the body, are critically dependent on the environment in which they live. And that subtle degradation of this niche, the environment where the stem cells lives and the signals that it receives, actually contributes contributes to the loss of the stem cell function. A good example of this is the thymus. The thymus is an organ that we all carry from childhood behind our sternum, right in front of the heart. It's one of the major sites for T cell production, and it's one of the earliest organs to involute. And so by the time you reach 50, 55, most of us actually lost our thymus, which we think is one of the reasons why we become less and less able to mount an effective immune response as we age. And there's growing evidence there that the loss of this niche, the thymic epithelial, is actually what's causing this.
So let's go to Lloyd Mitchell. Lloyd?
Hi, can you hear me?
Yes, loud and clear.
Hi. Mutations in a gene called lamin A cause progeria, which is a rapid aging syndrome. And researchers at NIH have shown that normal people also missplice this gene at a lower rate than the progeria patients because it's a mutation that causes a splicing error. So I was wondering if there's been further evidence, or if you believe that missplicing of lamin A plays a role in normal aging.
That's a good question. I'm aware of this data. I do not have an answer. There is growing evidence that one of the defects associated with aging and cancer as well is misfiring of splicing. Splicing that does not occur as it should happen. And I think it's partly part of this large picture that we talk about. Maintenance of your proteome is dependent on efficient and correct splicing of every protein. So I certainly can. It certainly makes a lot of sense. One thing that I would say is everyone loves the idea of a single theory for aging. And I always point to the fact that aging is a multiform event, and this type of lamin A missplicing is certainly could be one of the contributing factor. One of the hard thing that there was a famous paper published a number of years, close to 10 years ago, called The Hallmarks of Aging, which identified a series of misfiring or malfunctions that happen, molecular defects that happen during aging. This included stem cell loss. It included loss of proteostasis, the ability to fold your proteins correctly. It included loss of mitochondrial function, and so on. What the field is at the junction right now trying to build a hierarchy so that we have a little more of a unified model of what's happening during aging. And I suspect that there has to be some higher level order that we can actually identify, because identifying the hierarchy in terms of all of these abnormalities that occur during aging is going to be really critical. It could be that lamin A missplicing is going to be one of the key factors. At this point, we study all of these abnormalities, including loss of stem cell function. We don't have a good sense of what are the ones that are really the most important. What comes first? What should we be target first to have the maximum effect? I hope this answers your question.
Thanks very much.
We have a question from our computer whiz and PSW member, Jared McQueen. He wants to know, what do you think the best fidelity market available NAD supplement would be? I'm sorry, the best high fidelity market available NAD supplement would be. Do you yourself use the supplement? And what do you think the recommended dosage for an average size adult would be to see the healthspan longevity benefits? He's currently taking between 600 and 900 milligrams of nicotinamide riboside.
Very good. It's a good question. I usually don't share what I take in public, but since he shared it, I'll share what I take. So I have, as a physician scientist, I've spent my whole life actually self-experimenting on and just really looking at biomarkers and looking how I'm feeling, how I'm performing in terms of sports and the quality of my sleep and so on. And I have settled on NMN. Now, it turns out, NMN, at least in my case, I clearly see a difference when I am taking it. The problem about NMN is that there are many forms on the market. Many of them are not clean or pure. And I think, while I cannot really advertise on a specific companies here, I'm happy to have this conversation in private. There are a lot of products on the market, especially if you go into the supplement market. And I would urge caution in terms of what you're buying, because, for example, we found a significant fraction of the NMN that is sold on Amazon is contaminated with LPS for the biologists. So LPS is a bacterial product. It's also a product that causes inflammation. It activates so-called toll receptors. And I worry about people who take NMN hoping to do themselves good and actually are eating a product that is contaminated with a bacterial product. The reason it's contaminated with a bacterial product is that the synthesis of NMN, at least by some companies, involves the use of a bacterial kinase, which is not very pure. So personally, I take about 500 milligrams of NMN every day. And I have some colleagues who are taking up to a gram. I worry a little bit about going to these ranges because obviously the higher amount you're taking, the higher the effect, but also the higher potential for complications. And I always, when people ask me, how do you choose your dose? My answer is the better is the enemy of good. So if I feel an effect at 500, I think I keep it there.
Well, thank you for sharing that. We have a question from Rob Klein. Rob?
Oh, hello, Victoria Hutter.
Good.
Yes.
Thank you.
Sorry. I was just interested in whether you could speak a little bit more about the food as medicine that was in one of your slides.
Yes, yes, food as medicine. So I'm passionate by this area, along with exercise. I think I really believe if you want to attain optimal health today, based on what we know, these are, along with sleep, these are the three major areas that you should focus on. I'm very interested in modalities of fasting. So I would say that the data on, should you eat low fat versus low carbohydrate versus low protein is not convincing. And the same is, should you eat a vegetarian versus vegan versus an omnivorous diet, which one is the most healthy? The evidence there is, again, not convincing. There are people who attain really great health on eating meat, and some people who, so do it on vegetarian. I think it does not appear to be the key factor unless you go to extremes. There's growing evidence, and to me this is a source of worry, that a lot of people are eating excessive amount of proteins. I am disturbed by my visits to a Whole Food market and looking at the number of enormous bottles of protein, whey protein, and I'm always wondering who is eating this instead of eating a normal source of protein, which would be a normal diet. So there is evidence that excess protein intake is associated with increased cancer risk, and that mild protein restriction might actually be protective. But this is only true until the age of 70. It turns out if you are older than 70, a little supplementation with protein might actually be beneficial. So these are all really complicated questions. It's hard to make single recommendation. I would say having a fasting protocol in your lifestyle, I think, is something really beneficial for many people, and especially not being overweight. Beyond this, I think I'm happy. This is probably about an hour. I'm happy to come back and give you an hour lecture on what we've learned about the role of fasting. Fasting really activates a lot of incredibly exciting pathways. I think it's been part of our many of our cultures, mostly from religious cultures, have a fasting at least a year. I'm thinking about Ramadan and Lent and all this. So I think we are rediscovering many old traditions and the wisdom of our forebears, and I think I'm quite excited by this whole field of research.
Well, we'll keep in mind your offer to come back and give another talk.
Okay.
We have a question from Mike Cohn, another PSW member and a statistician. Mike?
I'm sorry, I missed your question.
Is curcumin in turmeric? And if so, is turmeric a good thing to take?
Again, I will put it in the group of the supplements. Not proven, suggestive, but not proven. And probably one of the key questions there lies in the quality and the bioavailability of the supplement. A lot of work is conducted with these bioflavonoid and other compounds like curcumin, and many of them actually do experiments in which they are injecting these compounds in mice and seeing all kinds of interesting effects. So the literature on turmeric and curcumin are provocative and interesting, but it's very different to go to humans and actually to expect this to be absorbed when ingested orally. So there are lots of different forms, there's different quality. I would say, again, it is of the order of self-experimentation. If you have the feeling it's making you feel better or it's changing some aspect of your physiology, even if it's a placebo effect, I think one of the good things about many of these supplements, at least when consumed in regular doses, are not toxic. It becomes different when you increase the concentration and the amount that you're taking in semi-pharmacological doses. So it's in some way, this is the dilemma of this whole field of supplements. It is a huge, $50 billion industry in the US, unregulated. Beware of what you're buying. Try to buy these supplements from, there are a number, I would say a handful of companies that actually have high standards in terms of measuring impurities and measuring actually that what you think you're buying is actually in the product. So there's lots of literature on this topic, and I invite you to, self-experiment, but always with a critical eye.
So we have a question from the web, and we'll take a couple more questions and then we'll close out. But the question from the web is from Jack Mooser, and he wants to know about sleep apnea. How significant is sleep apnea in determining lifespan? And is there evidence that a CPAP machine makes a difference?
The answer is yes to both. Sleep apnea is a very significant risk for early death and for all of the complications that we've talked about. I mean, one of the things that is hard to determine is whether quite often sleep apnea is associated with the weight excess. And so it is not always easy to differentiate the two, but it's clear that disrupted sleep does not help and is really puts you at risk for these conditions. The CPAP machines is the best treatment that we have today, short of surgery in some cases when there's a septal deviation or other abnormal formation in the nasal cavity. So yeah, the CPAP machines is clearly, at least for the people I have discussed personal experience of using them, it is a life-changing condition.
So we have another question I'm going to take from the web. Yij asks, concerning exercise, he used to get a full shift of exercise at work, but his doctor said that's the wrong kind of exercise. He would like to know if there's a difference between physical labor and healthful exercise.
No. Physical labor is a form. in some way, this is why I'm not crazy about the term exercise, because actually it is not, you don't need to exercise, you need to be physically active. The most famous study in the whole field on the effect of physical activity and health was actually one that was conducted by comparing the health trajectories of London bus workers. And if you've been to London, you know there are on their buses, they have two conductors, one who actually drives and the other one who goes up and down the stairs all day long. It turns out, so the first study looking at the effect of physical activity compared their life trajectories. It turned out that the conductor who was actually driving and sitting all day long had a high rate of heart attack, neurodegeneration, all of the complication of aging, versus the one who went up and down the stairs actually was relatively protected. And so that's the best demonstration. It's not necessarily exercise, it's the type of physical activity that you do all day long. It's been also said that what is really toxic is prolonged sitting in a chair or a couch. And so there's a lot of interest now in creating working environment where people are encouraged to walk 5 minutes, every 30 minutes. So being completely inactive is really one toxic aspect. I also, I love the data showing the effect of walking on health and longevity. Several members of the audience might have a Fitbit, which will tell you that you should be walking 10,000 steps a day to be healthy. And so I was always perplexed by this. I could not figure out where this number was coming from. It is 10,000 steps is actually quite a bit. And so what happens, a lot of people get their Fitbit, they walk 10,000 steps for the first 2 months, and then they get discouraged, and then they don't do anything. So someone, actually a colleague, conducted a study looking at the effect of the number of steps that you walk in a normal day versus your longevity or your risk of heart disease and cancer and so on. And it turns out that you get 80% of the benefit of walking by walking less than 5,000 steps a day. So obviously you get a little more, 80% of the effect is acquired during the first 5,000 steps. And 5,000 steps to be, as I mentioned earlier, is a 30-minute brisk walk, which I think everyone should include in their health routine every day. You see, with these types of physical activity, you see a 40% decrease in cancer, 40% decrease in heart attacks, a significant decrease in neurodegeneration, and the list goes on and on.
I'll start walking right away.
If you're not walking today and you want to do something to help yourself, I would say that's number one.
Well, my wife has been urging me, so. Last question, and I apologize to those of you who have not been able to ask a question. We're just over our time limit here. So, Ehrlich, you get the final question.
He has a question. It says, to what extent has the research on Dolly the sheep informed this field, the fact that she was born older? I can try to answer this if you're interested. So this whole idea of one of the most remarkable findings, so Dolly the sheep, I won't be able to talk about her age because I'm not aware of this, but one of the most remarkable aspect of aging is the fact that it is reset at each generation. So when the egg is formed from the father and the mother, the whole process gets reset perfectly. So when as we go through life, as generations go through life, there is not a progressive, slowly. You could imagine that the process would not be reset perfectly, which would lead that every generation people would get a little older sooner. We don't see this. The process is actually reset perfectly. So this has led people to say that maybe if we could help the organism to reset itself to this stemness, we could maybe suppress aging. And there are groups that are actually now pursuing the ability of factors that increase the stemness of organs try to reset the aging process. And there was one paper published that showed that they could do this with genetic manipulation in mice. There's a lot of research right now pursuing this idea of epigenetic reprogramming as a way to reset the aging process. I would say it's way too early for humans, but these are some of the experiments that are currently underway in animals, dogs, and so on. So stay tuned. I think this is a bit fringe, but I think it's quite exciting in terms of what it looks like could be done in the future.
Well, this is probably not apropos, but many years ago, Nature on April 1st, or their issue near April 1st, will publish a April Fool's report. And 1 year, when I was just starting out as my law career, they published a News and Views, which I took completely seriously. And the experiment reported on was shotgun cloning DNA from carp, which are very long-lived, supposedly immortal, into mice. And it reported that the mice these mice that were cloned from the carp DNA, that some of them didn't age. They just went on and on and on, and then they fell over dead after 10 times their lifespan. And it was really a great April Fool's. It took me in completely.
But so I can tell you, I can answer to this if it's a bit of a question, that there are some areas of investigation in this field. Now, one thing that is remarkable about aging is if you compare different species, it seems that the smaller you are, the shorter you live. So if you look at mice living, basically 3 years versus the whale living 200 to 250 years. So you can actually draw a regression line that plots the weight of an animal and its average lifespan. And most animals actually follow that line. What's remarkable is that there are some incredible exceptions. And I think these are the ones that are really interesting. I'll give you a good example. If you look at an animal which is the size of a mouse, which would be a bat, bats are, will weigh about the same weight as a mouse, 20 grams, but they live 30 to 40 years. And so do birds, who are about the same size. Look at a parrot, is about the size of a rat. A rat lives 3 to 5 years. A parrot would live to 70 years. So there's a lot of interest right now understanding what are the genes that define these gain in life expectancy. And I know that some colleagues have actually proposed to replace, there's another animal called a naked mole rat. Some of your audience might have heard about. This is a small rodent, which is about the size of a big mouse, and it lives about 30 years. So there are some efforts now to replace every target genes in the mice that with the corresponding ortholog from the naked mole rat. And I think these are things that are going to be really interesting in the future, but we're not there yet.
Well, I have to thank you very much, Eric, for giving this very informative lecture and for seeing it through all the questions and giving quite thorough answers. And we probably will be sending you an invitation to come back and talk about fasting.
In person the next time.
Yes, and by the way, you do get a rain check. So if you're in DC,
all right,
we'll have at least a dinner and maybe a little reception for you to make up for this having to be done by Zoom.
It was my pleasure to be here. I noticed that we still have a really good audience. I appreciate that they stuck with us until the end, and it was a great opportunity for me, and I'm delighted to have met you all.
Well, thank you so much. So thank everyone for joining us. The recording of tonight's lecture will be available to everyone on the PSW Science YouTube channel, where you'll be able to see it anytime you want. Please share the link with your friends and subscribe to the channel for notifications on new postings. Before you go, we have a few announcements. There's been a change in the schedule. Our next lecture will be in 2 weeks on October 23rd. The next speaker will be Prasad Sinisi. His lecture will be on the mathematics of voting and electoral systems, such as the Electoral College and the rank order voting system that's been instituted for the primary elections in California. The results of mathematical analyses of such systems sometimes produces highly counterintuitive results, which makes them especially interesting. Prasad is a professor of mathematics at the Catholic University. Kuo Kwang Ni, who was originally scheduled for this date, will be speaking to us on a later date to be announced shortly. The schedule for the spring has been posted to the website. Please check there frequently for updates. Last but not least, let's thank tonight's crew. James, Robin, and Ann for doing a splendid job of keeping the show running smoothly and monitoring the chat on YouTube. And with that, I will now adjourn the 2426th meeting of the Society and wish everyone a good evening. The meeting is adjourned.